endoplasmic reticulum in the cell is like the highway through the cell. It's the highways and byways through the cell. It forms a channel or a pathway through the cytoplasm. It gives the things inside the cell a way to travel through the cell. Two forms of endoplasmic reticulum. If ribosomes are attached to the endoplasmic reticulum, we refer to it as rough, because under the microscope it looks like sandpaper. If the endoplasmic reticulum doesn't have any ribosomes, we refer to it as smooth. So this picture is just showing you a ribosome, and this is showing you the smooth ER and the rough ER. So if I were to ask you what organelle is involved in protein synthesis, and your choices were nucleus, mitochondria, rough ER, or smooth ER, what would you answer? Rough ER. Why? Because it has ribosomes attached to it. The Golgi apparatus is kind of like the UPS store for the cell. The Golgi apparatus takes the proteins, finishes packaging them up, and then ships them out. So this I always call the UPS store, right? What do you do at the UPS store? You take your stuff, they pack it up, and they send it somewhere for you. That's exactly what the Golgi does for the cell. And then lysosomes are the housekeepers, right? Think of Lysol. You clean your house with Lysol. Lysosomes are the housekeepers. They clean up any cellular debris. They're little organelles that are filled with very powerful digestive enzymes. And those digestive enzymes clean up all that cellular debris, all kinds of waste and debris that are in the cell. Cytoskeleton. This is the skeleton of the cell. And it basically is a bunch of rods and tubes that give the cell shape, and to a certain extent will help with cellular movement. Picture up here, it looks like broken spaghetti. That's the cytoskeleton. And then centrioles. We're going to talk more about centrioles towards the end of the chapter, but basically centrioles are involved in splitting of DNA for reproduction. Alright, so we're going to go back to the cell membrane. So a couple things to remember about the cell membrane. It's semi-permeable. That means certain things can get in and certain things can't. Certain things get out and other things have to stay in. So that's an important feature of the cell membrane. And also, it's made up of the lipids and the proteins. So we have a double layer of lipids and then proteins poking throughout. In order for the cell to function, things need to be able to get into the cell and other things need to be able to get out. So things that we need in order for this cell to survive, things like oxygen and glucose, they need to be able to get into the cell fairly easily. Things that the cell needs in order to perform its functions, like amino acids, also need to be able to get into the cell so that they can perform whatever function they need to do. In the process of the cell function, the cell's going to produce waste products of metabolism. So all that stuff needs to get out of the cell. There's two mechanisms that allow things to move across the cell membrane. We divide those processes into either active transport or passive transport. So the first one we're going to talk about is passive transport. And passive simply means it requires no ATP. It requires no energy. The cell doesn't have to expend any energy to bring stuff into the cell. And there's 4 types of passive transport we're going to discuss: diffusion, facilitated diffusion, osmosis, and filtration. Active transport requires ATP. It needs energy. The cell must expend energy in order to accomplish an active transport. So 3 types of active transport we're going to cover are active transport pumps, endocytosis, and exocytosis. So we're going to start with passive transport, and passive again means what? No ATP, no energy needed on the part of the cell. Okay? We have 4 different types of passive transport. The first one is diffusion. And diffusion simply means the movement of substances from an area of high concentration to low concentration. When the concentrations are equal, it's said to be at equilibrium. Example in the book talks about the movement of oxygen and carbon dioxide between the blood and the lungs. So basically, when we breathe air in, there's more oxygen in our lungs than in our blood. So the oxygen moves from our lungs and into the blood. When we exhale, there's more carbon dioxide in the blood than in the lungs. So it moves from the blood into the lungs, and then we exhale it and get rid of it. Now, in some cases, we need a helper molecule. And in that case, we call it facilitated diffusion. So facilitated diffusion is very much like plain diffusion. Things are moving from higher concentration to lower concentration. But instead of just moving independently, the molecules need a helper. So the helper molecule increases the rate of diffusion. It helps it. So if you think of rolling a ball down a hill as diffusion, you're going from high to low. If you take that ball and carry it down the hill, now it's facilitated diffusion. The ball is not expending any energy, but he needs a helper to get down the hill. So the key with facilitated diffusion is the presence of a helper molecule. Okay, so osmosis. And it's the movement of water or the diffusion of water through a semi-permeable membrane. So some things can get through, other things cannot. When substances are confined in a small area, they want to pull water to wherever they are. That force, that pulling, is called osmotic pressure. So for example, if you have a solution in the beaker, and the beaker is separated by this selectively permeable membrane. That means only water can get through. The little dots cannot. You're more concentrated over here than you are over here. So water wants to go where the party is. Water wants to go where all the particles are. So water gets pulled from this side over to this side. And it's going to keep getting pulled over there until the concentration on each side is the same. I said osmosis is defined as the diffusion of water. Diffusion goes from what to what? High to low. So in this container, where is there a higher amount of water? Over here, right? Because there's fewer particles, so there's more water. So the water is going to go from where there's more water to where there's less water. So you can look at it either way. You can look at it as the particles pulling the water or the water going to where there's less water. Now, on the same line as osmosis, we're still talking about osmosis. Talk about this concept of tonicity. And basically, it's going to describe where the water is going to move. And here we're talking about movement across the cell membrane into a cell. So we have isotonic, hypotonic, and hypertonic. So if I take a blood cell and I put it in a solution, and that solution has the same exact concentration as the inside of my cell, we say it's isotonic. Iso means the same. So it's the same tonicity as the cell. Red blood cell has a sodium concentration of 0.9% sodium. Normal saline has a concentration of 0.9% sodium because that's what a blood cell contains. So normal saline is isotonic to blood cells. All right, so if these little black dots represent sodium, then the concentration of sodium outside the cell is the same as the concentration of sodium inside the cell. But now look what happens if I put the cell in a different type of solution, a hypotonic solution. Hypo means what? Hypo, below. So now I'm going to put the cell in a solution that is less concentrated than the cell. It's going to look like this. The cell is highly concentrated. The solution is less concentrated. So where does water want to go? Water wants to go where the party is. So water wants to go inside the cell because that's where the concentration is. And then what happens to the cell? The cell starts to gain water, gain water, gain water, and it starts to expand. And eventually, it's going to burst or lyse. Now, I put a picture of a hot dog up here. When you take a hot dogs are full of sodium. Take one of those and put it in a pot of water and then heat it up. What happens to the hot dog? It expands and eventually it will burst through the little casing that it's in. It expands because the water is moving into the hot dog. There's more salt inside the hot dog than there is in the water around it. So the water is hypotonic the hot dog. So where's the water going to go? Inside the hot dog will swell and eventually rupture. And then finally is a hypertonic solution. In a hypertonic solution, the solution is more concentrated than the inside of the cell. So the cell has only a few particles. Whatever solution it's in has a lot of particles. And water wants to go where the party is. Water wants to go where the concentrated particles are. So water gets sucked out of the cell. So the cell will shrivel up. And that shrinking of the cell we call crenation, or the cell will crenate. Now filtration is when water and substances move in response to pressure or a push. They need some kind of push. Okay? Again, they're not expending any energy, but something is pushing them. So I put coffee filters up here. So think about coffee filters. If you put the coffee grounds or the coffee beans or whatever in the filter and you pour hot water over it, what comes out of the bottom? Coffee. The coffee's not moving because there's a higher concentration up here and a lower concentration down here. The particles of coffee are pushed through the membrane, which would be the filter, because the hot water is pushing them through, not because there's a concentration higher up here and lower down here. The picture in your book shows you the picture of this syringe. You press the plunger of the syringe, the water's going to come out through the needle. If you poke holes in the barrel of the syringe and push on the plunger, then the water's going to come out any of those holes. Again, because it's being pushed from behind. Same thing happens in the blood. Water and particles can get pushed out through the walls of the capillaries. There's very small holes in the capillaries. And water and dissolved substances can get pushed through those holes. The pressure of the heart pumping is pushing the blood through those capillaries. When we get to renal, we're going to talk about filtration again.